basically is covering, I was assigned the melanocortin 1 receptor for my protein. So that today I'll talk a little bit about that, and I will include the 3D models and give an explanation for those as well. So this is just the portion where I'm going to show my face, because I'm going to turn the camera around so that you can see both proteins and the mutation. Okay, so for this clip, I am including the first structure, which is just the original amino acid, and that includes arginine. Okay, so I have some labels going on here. So you can see here is the inside of the cell, and then here the red strip, I use that as my cell membrane, and the outside of the cell is just above the red strip. And then you also have the green rope-like structure, which is the side chain. And then here's just a full little glimpse of it. Okay, so here I have the new mutation amino acid, and this one includes cysteine. So really it's just the same as the last one as far as things are placed, except we have this new little thing right here, and that is cysteine. And so that is just the new mutation, which is the difference between the first amino acid and this one here. But even though they're the same, I will still go through and tell you all of my labelings. So of course at the very top you have the outside of the cell. This red strip, I consider that to be the cell membrane. And then here is the inside of the cell, where you also can see the cysteine at. And then the green thing is the side chain. I just want to go ahead and give a full glimpse of both new amino acids. So here is it from the top, and then here when you have it, you can just see all the labelings. Okay, so I brought back amino acid cysteine just so you can see everything. Has a really nice view from the top. And then you can just bring it back down to see labelings. Okay, so now I have both models side by side, and I'm just gonna briefly discuss a little bit of information about them. Okay, so first off, as I stated in my introduction, the protein that I was assigned is the melanocortin 1 receptor, and my mutation is arginine number 67 is replaced by cysteine. So some normal functions of this protein is it is involved in creating a normal pigmentation. To be specific, the melanocortin 1 receptor is involved in causing the pigmentation of skin to be regulated. It helps with the risk of melanoma and keeps ultraviolet light response under control. I would categorize melanocortin 1 receptor under receptors, of course. This protein can normally be found in the upper exterior of the melanocytes. This protein can usually be found in organisms such as animals and humans. So the protein becomes less active because the mutation causes the melanocortin 1 receptors to lose the ability to bind the AMS. This causes eumelanin to not produce anything, and it alters the pigment's pathway. Okay, so for this section, we're going to focus a little bit more on the original amino acid, which includes arginine. So some properties of the original amino acid is this is a hydrophilic group. I would characterize it as short in its structural form, positively charged, and I think it's kind of coiled just because of that up there. And I'd say it's average size in comparison to other amino acids. When discussing the type of bonds the original amino acid can participate in, I'd say arginine side chain contains water and it associates with other water-soluble and polar side chains. Okay, so now we have moved on to the new mutated amino acid side chain, which includes cysteine, and some properties of this new side chain are it contains hydroxyl, carboxyl, and sulfhydryl main functional groups. So the new mutation is polar and it's smaller in size compared to the original mutation. This mutated side is non-charged, or also known as neutral. So the new mutated side is able to create cross-links or disulfide bridges amongst the sulfhydryl groups that help and stabilize the protein structures. Alrighty, so we're almost at the end of this presentation, but we have a few more things to discuss first. So I was asked how the mutation affects the protein function, and I feel that the mutation affects the protein function by causing eumelanin to no longer be able to be produced. It also changes the direction of the pigment. The function of the cell will be altered and decrease the stimulation of protein and stops other proteins from being expressed. So some limitations that I had with this project would have to be not being able to represent key factors of the actual protein, such as alpha beta links, functional groups. I can talk about these things, but you can't really see them, and bonds. So I'm pretty sure I discussed them all throughout the presentation, but you're just not able to see them with this model. So yeah, this really sums up my presentation. Thank you.